Hey everyone. In this video, we are going to be talking about um, phospholipids, excuse me. And so in this video, the topics will be phosphoester functional groups, amino alcohol molecules, and then we'll look at two types of phospholipids, glycerophospholipids and sphingomyelins. And by the end of this video, you should be able to understand the different types of phospholipids and what they're used for. Let's talk about phosphoesters. So phosphorus atoms can form phosphoesters, similar to how carbon atoms form regular esters. And so a normal ester is when you have a carboxylic acid and an alcohol, and you kick out H and OH, which leaves as water, and it gives you the ester, right? That's your ester functional group. It's your esterification reaction. We can do the same thing, but instead of a carboxylic acid, we use phosphoric acid. Phosphoric acid, remember, was H3PO4. So you've probably seen it as H3PO4, but maybe you never thought about its Lewis structure. But it can do the same reaction here, where we're going to kick out H and OH. That's going to leave as water. And what we have now is we have a ester, but with a phosphorus atom instead of a carbon. And so we call this a phosphoester. Okay. Now, you can also have a phosphodiester. Because phosphoric acid has OH on both sides, we can kick out a water molecule on both sides. And so we'll lose two water molecules. And what we have here is a phosphoester on that side and another phosphoester on that side. And so since it's two phosphoesters connected to the same phosphorus, we call it a phosphodiester because there's two of them. So phosphoester or a phosphodiester. And this functional group is not talked about in your book, but this is the name of it, and you're going to be seeing it a lot. So I thought it made sense to go back and fill in this missing void where they didn't talk about it in chapter 12. The next thing I'd like to talk about are amino alcohols. So an amino means that it has an NH2 group or like a nitrogen group. And an alcohol obviously is an OH. And so an amino alcohol has both functional groups in the same molecule. And so there are three common amino alcohols that you see associated with phospholipids: ethanolamine, choline, and serine. And they all kind of have that similar core structure here, where you've got two carbons, OH, and a nitrogen. Two carbons, OH, and a nitrogen. Two carbons, OH, and a nitrogen. The only difference is what else is attached. So ethanolamine is like the bare bones. Choline has three methyl groups attached to the nitrogen. So technically it's a quaternary ammonium, but technicality. And then serine has this extra carboxylic acid sticking off the side. But they all have that same core amino alcohol structure. So let's talk about phospholipids. Phospholipids are very similar in structure to a triacylglycerol. So triacylglycerol has the glycerol and then the three fatty acids. A glycerophospholipid has glycerol, two fatty acids, but then it has a phosphodiester with one of those amino alcohols sticking off. And then sphingomyelins have this unique molecule called a sphingosine, which we'll talk about in a little bit. It's got one fatty acid, and then it's got your phosphodiester with one of the amino alcohols on the end. So triacylglycerol versus a glycerophospholipid versus a sphingomyelin. And we're going to look in detail about these two types of phospholipids. So glycerophospholipids resemble a triglyceride, except for on the third spot, instead of another fatty acid, we've got the phosphodiester with the amino alcohol. And so here's the structure, just abbreviating the rest of the molecules are. These would be your two fatty acids, and this would be in blue, one of these amino alcohols. Now, there are two main types of glycerophospholipids, lecithins and cephalins. And the only difference between a lecithin and a cephalin is which amino alcohol is attached. If you use an ethanolamine, it's known as a cephalin. If you use choline, it's known as a lecithin. That's the only difference. And the particular fatty acids that you use here don't matter. It could be any fatty acid, but as long as it's got the phosphodiester with ethanolamine, it's a cephalin. Phosphodiester with a choline is a lecithin. And so we often abbreviate the structure of a fatty acid in this cartoon form. Now, at physiological pH, these nitrogens exist in the charged state. So when they're dissolved in water, this actually exists as NH3 plus. This one's already positively charged because it's got 4 bonds, and this one would also exist as the NH3 plus. So in your body, when they're dissolved in water, these nitrogens are positively charged, as shown here. But on a phosphoric acid, when this is dissolved in water, it loses these hydrogens and they exist as an O minus. And so you have these ionic charges, which makes this region of the molecule very, very polar. This is what we call the polar head region. And then we have the nonpolar tails. So polar head, 2 nonpolar tails. And so we abbreviate it like a little 2-tailed sperm here. This is the polar head. These are the nonpolar tails. We will see this cartoon diagram in a future video. So let's talk about sphingomyelins. So sphingomyelins contain a sphingosine backbone. They contain a fatty acid connected as an amide. And then we have a phosphodiester with one of the amino alcohols. So sphingosine, as highlighted here in green, looks like this. It looks similar to glycerol, sort of. It's got 2 alcohol groups, but then it has an amine group. And then it's got its own long carbon tail as part of the backbone. So that's a sphingosine. And so we connect a fatty acid to the nitrogen here as an amide with the fatty acid. And then on the third alcohol, we connect the phosphodiester with one of those amino alcohols. And so this is the general structure of a sphingomyelin. Now, why do we care about these? These are really important in your brain. So these are what form the white matter of the myelin sheath, which is the coating around your nerve cells in your brain. That myelin sheath coating, it increases the speed of nerve impulses, and it insulates and protects the nerve cells. There are certain diseases where the myelin sheath degrades and it leads to dementia and other neurological conditions. So it's very important to have sphingomyelins being healthily made in your body because you need them for your brain. And so here's an image showing your neuron. And here's the axon of the neuron. And surrounding it are these coils, these coatings of sphingomyelins. And they form like they're blue in this picture, which is kind of silly because they actually look white. So if you look at the brain under a microscope, like it just looks white. And it's the white matter. It's the myelin sheath formed from the sphingomyelins. So, once again, to recap this video, phosphoesters and phosphodiesters are formed from phosphoric acid. There are 3 different amino alcohols. A glycerophospholipid has glycerol, 2 fatty acid esters, and then the phosphodiester with the amino alcohol. Lecithins and cephalins are the 2 types of glycerophospholipids, depending on if you use ethanolamine or choline. And sphingomyelins have sphingosine, 1 fatty acid attached as an amide, and then you have the phosphodiester with the amino alcohol. So that's the structure of the 2 types of phospholipids. Thanks for watching. See you in the next video.